Hello, Assalamualaikum warahmatullahi wabarakatuh. My name is Satia Putra, and in this video, you'll see some explanation about enzyme.
Enzymes.
Hey, Professor! Just the other day, I saw this small man swallow an entire steak bigger than the size of his head. And it got me to thinking, how could he possibly digest all that in just a few hours?
Well, the steak is first chewed in the mouth into bits and pieces that fall into the stomach. You might already know that the stomach contains a powerful acid that can dissolve even metals. But what you did not know is that this acid is not what ultimately breaks down the steak. No, it's very tiny and special molecules called enzymes that do the job. Strangely, these enzymes find the stomach acid a cozy place to work in.
Whoa! It's like small piranhas finishing up a whale in a sea of acid. Imagine what I could do with that!
Ooh. Huh?
Well, it doesn't work that way.
What do you mean it doesn't work that way?
I knew you were going to say that. To find out, you need to know what exactly enzymes are and how they work. Whenever you're ready, click Next to continue. What are enzymes? They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they would be too slow to support life. There are many different types of enzymes, and they come in all manner of shapes and sizes. For example, what you call the piranha enzyme is actually pepsin. It is only found in the stomach and digests proteins. In your mouth, there is amylase, which breaks down starch. In your cells alone are hundreds of enzymes that control your cell activity and defend them from invasions by microbes like bacteria and viruses. Though enzymes can appear very different, they all share a few characteristics in common. Let's now take a look at what these are. Number 1, each enzyme has an active site. This is the groove in the enzyme where the molecules they act on, called substrates, are captured and made to undergo a reaction. The substrates are either broken apart or combined together to form products. For instance, starch found in the bread that you eat is digested to form simple sugars called glucose that is then absorbed into the body. Glucose is used up as energy, but when there's too much of it, special enzymes combine the glucose molecules together to form a giant molecule called glycogen, which is then stored away in the liver and muscles for later use. Number 2, enzymes are very specific. Each enzyme can only bind to one type of substrate. Let's look at the case of lipase enzyme, which breaks down fats. Molecules like glucose and proteins cannot fit into the active site of lipase. Only fat cells can bind to the active site because they have the right shape. This ability to act on just fat cells is very important for lipase. Lipase converts fats into energy. Now imagine what would happen if lipase can also convert proteins into neurochemicals for the brain to function well. If you no longer need the extra energy from your storage of fats, lipase will be deactivated. But this means that lipase can no longer produce precious neurochemicals for your brain. In other words, when you stop burning fats, you end up damaging your brain. To prevent such freakish scenarios from happening in our body, enzymes must only act on one type of molecule. Number 3, enzymes are recycled. Enzymes are not altered by the reactions they speed up. Immediately after they finish one reaction, they can act on a new substrate. This way, only a small amount of enzyme is needed to speed up thousands of reactions.
Enzymes kind of act like a coupon. activation energy. All reactions cost energy to start. An enzyme acts like a coupon. If we use one for our reaction, we spend less energy. So here is a chemical reaction in your body. We have the starting energy, and then we have a high curve with our activation energy. If we can't afford the activation energy, we can use an enzyme to lower it. That's much better.
When enzyme and substrate molecules are mixed, they randomly collide, forming enzyme-substrate complexes. As temperature increases, so does the kinetic energy of the molecules, and therefore also the rate at which these collisions occur. There comes a point at which the maximum rate of reaction is achieved, and the temperature at which this occurs is known as the optimum temperature. If the temperature continues to increase beyond the optimum, the enzymes begin to lose their three-dimensional structure and become denatured, therefore reducing the rate of reaction. Now you need to be able to recognize the graph of the effective temperature on enzymes. It looks like this, with the peak being the optimum temperature. To explain up to the peak, as the temperature increases, so does the kinetic energy of the enzymes and the substrates, resulting in an increased frequency of collision between the two, thus increasing the rate of reaction. But beyond the optimum temperature, this continuing increase in kinetic energy results in a loss of 3D structure from the enzyme and therefore a change in its active site. If the substrate can't fit into it, the enzyme is referred to as being denatured, and this reduces the rate of reaction. Now for a look at pH. Increasing pH values are as a result of adding hydroxide ions, or OH-. Decreasing pH is as a result of adding hydrogen ions, or H+. Either of these can potentially alter the charges on the amino acids that make up the enzyme, and this can lead to the enzyme's active site changing shape because the bonding changes, for example, and that could denature it. Or it could mean that the substrate can no longer bind to the enzyme's active site, and so either way the rate of reaction is reduced. The graph for the effect of pH on enzyme activity is very similar to that of temperature, although you might notice a more symmetrical decrease in rate of reaction either side of the optimum pH. On to having a look at substrate concentration. When we change substrate concentration, we are assuming that enzyme concentration is remaining constant. Initially, increasing substrate concentration from 0 will increase the rate of reaction as the substrates randomly collide with the enzymes and the enzymes catalyze the chemical reaction. However, there comes a point where all of the enzymes' active sites are filled and the enzymes are already working as quickly as they can to catalyze the reaction. This is where the maximum rate of reaction is reached, and any further addition of substrate beyond this will not increase the rate of reaction anymore.
Competitive inhibition occurs when an enzyme encounters a blocker which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as the non-competitive blocker is bound to the enzyme.